It's my pleasure and honor to introduce tonight's speaker, Dr. Luke Miller. Dr. Miller is an associate professor of biology at San Diego State University. He went to school at UC Santa Barbara and got his PhD at Stanford University. Luke is a marine ecophysiologist and intertidal ecologist and is primarily interested in how ocean animals deal with the physical environment, including the weather and waves, as well as how they deal with each other via competition and predation. Much of his research has focused on the Pacific Coast rocky intertidal and estuary habitats from California to Alaska, but he has also worked on New England rocky shore communities, on Antarctic scallops, and in kelp forests. Luke was born and raised in San Pedro, more specifically at the Cabrillo Marine Aquarium after school and during the summer, as his mom, Dr. Suzanne Lawrence Miller, was the director of CMA for over 30 years. The title of tonight's talk is Prying into the Private Lives of Mussels and Oysters. Thank you, and please help me welcome Dr. Luke Miller.
No worries. All right, let's see. Can you all hear me in the room? No? Yes? Let's see if we can fix it. Oh, is it still muted? No.
Yeah. One, and then...
Or oh, wait, yeah. Did that start working? I'm 5, right?
You're 5.
I'm 5.
Okay.
Okay. Yeah. Can we hear me okay? Is that, maybe that's better when it's out under the, from out shadow under my,
Okay.
my
Good.
chin. Okay. All right, I feel like I hear myself here, and hopefully they can hear me at home. Hopefully the little gray box for the folks on Zoom isn't covering up too much interesting stuff on these slides. So, oh, yes, we need to click in. I'm just gonna make this a laser pointer as well and see if we can make it go forward. Oh, did this get shut off? There we go. Yes, so, yeah, hello. So my name is Luke Miller. As Julie said, I'm at San Diego State University. I've been there since about 2018, and I sort of put myself in these multiple boxes of studying physiology, studying ecology, and also studying biomechanics. But most of my work has really been focused around shoreline and near shore critters, mostly invertebrates, mostly things like mollusks and arthropods. So I've worked all up and down the West Coast. I'm going to talk about projects from a few of these places. But as Julie said, I really sort of got my start right here in San Pedro. So here is 1989, the summer docent class. I'm the skinny kid in the blue circle over there. A couple of our audience members are in this photo as well. This is sort of where I learned public speaking, which I still am putting into practice to this day. The other reason I'm here is these two people on the left, my mother, Suzanne Lawrence Miller, my father, Alan Miller, who taught at Cal State Long Beach, and so he was the sort of threatening aura for a number of people in this class when they were in this audience when they were taking classes. And you know, like Julie said, I was here from the very, very start. You can tell it's the early 80s. I've got short shorts on and some striped socks holding my little balloon. This is around the time, I think, of the actual opening of the original iteration of this building here. My younger brother is there in the carrier as well. So I'm going to start by sort of talking a bit about sort of how I got to where I am. Part of that is, you know, starting out in San Pedro, moving to UC Santa Barbara for a couple of years, and then I spent my sort of formative graduate school years in the Monterey Bay. Stanford University has a marine lab in the Monterey Bay, on the Monterey Bay Peninsula. That little star there on the right-hand picture shows where that marine station is located, right next to the Monterey Bay Aquarium, if you've been to that location. I was there in large part because of my major professor at Stanford, who was Mark Denny. Mark Denny is a biomechanics sort of guru. That was his big thing. And biomechanics, sort of principles of engineering applied to mostly rocky intertidal habitats. He's sort of an eclectic person. You can see him standing there in the red jacket up on a wobbly wooden ladder perched on some rocks in the intertidal. He's pointing a camera out into the wave zone there because what this particular project was throwing balloons full of green dye out into the wave zone and then watching that dye spread and recording it and looking at what the waves were doing in terms of driving water flow close to the rocks, which is, of course, delivering food and delivering nutrients to all the organisms living on those rocks. You may also notice that down in the corner of that photo, there's the butt of a rifle. If you've ever thrown a water balloon into a body of water, you may have realized that they don't all, immediately pop and spread. And so one of Mark's graduate students, not me, had to sit out on the rocks with this air rifle and plink away at these dye-filled balloons until they've actually hit it and popped it and got it to start spreading. But Mark is sort of who got me into really doing this as trying to look at how organisms cope with their environment and look at it from a sort of behavioral and mechanical perspective. And then we sort of have layered physiology on top of that, because ultimately the design of organisms and their coping with the environment is all really physiology. Now, the wave-swept intertidal, the rocky intertidal, is a fairly dynamic environment. Obviously, you look out there and you see big waves and you think, oh my gosh, how do organisms hold on? How do they survive in those kinds of conditions? At high tide, obviously, crashing waves are a problem. At the same time, when the tide goes away, you've got a different set of problems. As a marine organism that needs salt water to live, it's probably where you get most of your food, it's where you cool off. When that tide goes away and goes out, now you're sitting in the air and you've become a terrestrial organism for a couple of hours. You may be baking in the sun, you may be freezing at night without that sort of moderating influence of the ocean water washing over you. And so those are the sort of environmental fluctuations that really are driving interest in sort of how do these organisms tolerate things. So I'm going to talk mostly about bivalves tonight. When I say bivalves, I think most people immediately go to dinner. Whether it's oysters or mussels, this is what I often think about as well. These things taste good. But of course, the unifying feature here is that these are bivalves. They have two shells. So inside those two shells is all the tasty stuff. On the left here, we've got a California mussel, Mytilus californianus, split open. You can see the gonads. Some of the gills are sort of folded up there. The mussels are the white stuff. The mussels with a C are the white stuff inside the mussel with two S's. And these things are really good sort of habitat formers themselves. So they grow in big aggregations. The photo on the right there, you've got one little mussel that's laying out its individual byssal threads onto all of its neighbors to anchor itself into this larger bed of mussels. And this is how they sort of hold on under those crashing waves. That characteristic of, sort of growing in big aggregations is often important because they are contributing three-dimensional structure for a lot of other organisms to live inside. They're also filtering the water, they're pulling phytoplankton out of the water, they're sort of cleaning the water in some sense, and really being fundamental, important parts of a lot of near-shore coastal ecosystems. I'm going to start actually by a little story involving a bivalve that doesn't grow in big beds connected to each other, actually scallops here down in Antarctica. Now, scallops are sort of an interesting critter because they are one of those bivalves that can actually get up and swim around. And if you've never seen it, here goes a scallop about to take off. And you'll notice that that scallop is sitting there clapping its two shells together. And it's actually creating little jets of water being forced out of the shell through the rear where the hinge is. The diagrams here on the right-hand side are showing you a scallop that is open and then a scallop that is closed, and the blue arrow represents water jetting out past the hinge of that scallop. And that is how they get themselves up and start swimming away when they are threatened by predators. We were interested, in the case of the Antarctic scallop, in how they actually manage to maintain that ability to get up and swim in these very cold waters. Because one thing about bivalves is that they have muscles to close their shells, but they don't actually have muscles to open the shells. And so opening the shells relies on basically having a little rubbery pad depicted by the white sort of shapes here near the hinge that actually is trying to force the shell open most of the time. And so in order to close it, they are fighting against that hinge. But in having that little resilient pad at the hinge, that forces the shell back open for each stroke, refilling the water as this scallop is swimming along. So to study sort of how the rubbery material that makes up the pad in the hinge of a scallop in Antarctica works under those very cold water conditions, we had to go ahead and go to Antarctica. So you leave from Los Angeles, you fly to Christchurch, New Zealand, you get on a big US Air Force cargo plane, which is not necessarily the cushiest kind of flying conditions, but they do move some airline seats into it alongside the helicopter that they shove in the back. You put on all your cold weather gear, you fly for a couple of hours down to Antarctica, you land on the ice. They groom the ice to make the runway there. And you have arrived at what in some senses just looks like a mining town. You know, it's just sort of a fairly desolate wasteland here with a whole bunch of ugly buildings on it. There are pipes running places. There's no trees. There's no green stuff. There's no lawns. There's no nothing growing out here because all year long it's really, really cold. But this is a basically science camp. The central building there is a whole bunch of fairly modern science labs, and they bring scientists down there to study all sorts of aspects of geology, climate change, ocean life in Antarctica. So we went down there with the hopes of getting our hands on some scallops and doing some mechanical stress tests on those animals. Now, the ocean around McMurdo Station often has a whole lot of ice on top of it. And so these two photos are out on the ocean on the sea ice. And the year that we were there, the sea ice had not actually broken up and flowed away for about 3 years. And so it just sort of continued to accumulate over the years. And the two people on the right there are holding an ice drill that they've used to drill down through the ice. And notice that there's about 20 feet worth of ice drills stacked on top of each other. That's how deep the ice was at this point. So if you're going to go get animals, marine animals, out of the ocean, you need to get through that ice. So they will drive a tractor out there on that very thick ice with a big 3-foot diameter drill, and they will drill a hole down through that ice. And so the folks on the left there are sort of clearing out this dive hole in order to allow divers to go ahead and dive in that dive hole. So here we are, here I am, in a full body dry suit, dry gloves getting put on the right-hand side here, a rubber hood, a mask. The only parts of my skin that are really exposed are really my lips and a bit of my cheeks. And you jump in the water and those just immediately go numb, and you use your teeth to kind of hold on to that regulator and just keep going for it. So you jump in the hole and you descend through this tube, for in this case about 20 feet in this 3-foot diameter tube. You get down to the bottom and pop out into the open ocean, and all you've got above you is this sort of 3-foot diameter hole with a bunch of light streaming down through it. But because there's all that ice everywhere else with snow on top of it, there's not much light coming down. So it really quickly turns into a night dive, even though it's the middle of the day with 24-hour light because we're there in the Antarctic summer. So you go down to the bottom, and what you see is actually a whole lot of stuff that looks very, very similar to what you see if you're diving out here. So, you've got sea stars, you've got sponges, you've got a feather duster worm, we've got sea urchins there. We've got sea cucumbers that are just hanging out, collecting food, a sort of nudibranch in the white thing in the lower right there. Big Nemertean worms, those purple things there that have wrapped themselves around this large sponge. Giant sea spiders, pycnogonids, which we have very tiny ones here in the west coast of the US. This one is about as big across as your hand, but they're not all that threatening because they are just sort of these bundles of legs that aren't interested in attacking you at all. There's urchins that put on little party hats for some reason. They collect detritus and they just sort of hold it on top of themselves, I guess, to disguise themselves. And there are some cute fuzzy things there as well. So this is a Weddell seal. And I mentioned that they go out and they drill a hole for the divers to go down. They actually always drill 2 holes because this sort of thing happens. This is a Weddell seal that has taken up residence in one of those dive holes. And you can imagine if you go down through the dive hole and then want to go back up and there's a seal sitting in your dive hole, you're not really going to share that dive hole. So you've got to swim yourself over to the other dive hole and come back to the surface. So we did a couple of dives. We collected lots of critters. We got a hold of scallops. And our goal was to basically, again, look at what that little resilient hinge, this black thing in the right-hand picture, how it behaves under a variety of temperatures. Because the thought is that an Antarctic scallop sits in water that is between -1 and -2 degrees Celsius all year round. And that's sort of the conditions that they have evolved in. And you can imagine at really cold temperatures, rubber often gets very, very stiff if we're thinking about car tires, for example. The same sort of issue is going to happen with these naturally secreted resilient materials that the scallop is making. And so in order to be able to continue swimming under these sorts of cold conditions, the scallop has presumably tuned the protein makeup of these resilient hinges in order to be springy enough in cold temperatures. We also wanted to look at what happens when you change the temperature, when you raise the water temperature. Does this scallop's rubber actually get too floppy and compromise its ability to swim? So we ginned up some sort of, fairly back of the seat of the pants kind of scientific equipment here in a water bath where we could control the temperature that this little resilient pad in the shell hinge was sitting at. We pointed a laser at a piece of mirror here that was attached to part of a syringe that we glued to the remnants of the shell, and we would sit there and we would just sort of twang that syringe thing and watch the laser bounce up and back and forth. And the trace here on the right was a trace of that laser moving back and forth across the ceiling of the lab, giving us a measurement of basically the frequency at which this thing was bouncing as we twanged it. So we could do that at normal Antarctic ocean temperatures, and then we could raise the temperature and watch this thing actually sort of get floppier and floppier. And what we come to the conclusion is that these animals work all right currently in very cold ocean temperatures in Antarctica. But if you take an Antarctic scallop and move it into warmer waters, it wouldn't effectively be able to swim itself because it would be able to close its shell with the mussel and shoot water out. But the reopening wouldn't happen at an appropriate frequency. And so they wouldn't be able to beat fast enough to get themselves up off the bottom and swim away. And so they would sort of just sit there uselessly on the bottom, closing the shell once in a while and not really going anywhere. So this is an example of sort of taking a mechanical property of something that an animal has created and looking at how it's impacting that animal's ability to survive. I then want to now return to sort of warmer climates here. A photo here from Central California, from Monterey. And a lot of what I do is looking at how stress impacts the survival, the growth, the performance of intertidal organisms like this bed of mussels here, and also look at how much that stress varies over various spatial scales. So if we look at a couple of animals that are just a few centimeters apart, do they experience the same environment, or do they experience slightly different or very different versions of that environment? So, something like a bivalve is making a choice to go ahead and open when it's got water over it. They're doing that, pumping water into the shell, feeding themselves, breathing as well. And we can monitor that sort of thing using various tools. So in this case, this little mussel in an aquarium has a sensor glued to the front of it. You see a couple of wires through the glue there. There's also a magnet glued to the other half of that shell. And with that, we can actually measure when that shell is opening and closing, so that we don't have to have a video camera pointed at it all the time, but we could take this animal, put it out in the wave-swept environment, and connect it to a data logger on shore and keep track of what it's doing under all sorts of different conditions. And one of the beauties of working in the modern age is that there's a lot of great, cheap or free open source tools for designing your own sorts of sensors and your own sorts of data loggers to go with them. So this is an example of a custom circuit board that I put together to make measurements on a couple of different sensor types for mussels that we were going to put out in the wave-swept environment. So you whip this up on the computer, you send the file off to a fab that sends you back a stack of those bare circuit boards, you spend some time soldering them together, and you've got a fairly cheap but fairly robust little data logger that you can then put out in the saltwater environment. And you know what? If it leaks and it destroys the board, oh well, you lost $15 instead of losing hundreds or thousands of dollars worth of commercial equipment. So in this case, what we were doing was looking at how these mussels were coping with that, again, wave-swept environment, but also coping with temperature stress at low tide. So I've got a little diagram here of a California mussel, like you would find out on the open shore here, with 3 different sensors attached to it. First, a temperature sensor, we call it a thermocouple. We would actually drill a little hole through the hard shell of the mussel and poke the tip of this thermocouple in there, sort of poking the mussel in the side of its tissue and gluing it in place so that we can get an internal body temperature for these animals. We also put a magnetic sensor on the end of that shell, like I was showing you in the previous video, so that on one half of the shell, we've got a sensor, on the other half, we've got a magnet, and as the shell opens and closes and moves that magnet closer or further away from the magnetic sensor, we get a changing voltage, and we can record that as the shell opening and closing. And then we also put a 3-dimensional accelerometer and magnetometer on a different part of the mussel, the little yellow diagram in the lower left of the mussel there. And this is the sort of chip that your smartphone uses to decide what orientation you are in. So for instance, if you've got one of those apps that shows you the night sky and it knows exactly where in the sky you're pointing, it's using that kind of sensor to detect, gravitational acceleration, so which way your phone is tilted, but then also compass direction as well, using the magnetic fields of the Earth. So we can put all of that onto a mussel, like in the central picture here, and then create little sort of, I don't want to quite say artificial mussel beds, because this is a mussel bed composed of entirely live mussels that were collected from the field. Some of them we attached all these sensors to. Others of them we just packed on regular mussels with no sensors to build a sort of fairly dense bed of mussels. And all of this is on a big piece of plastic with the data logger boards in those waterproof boxes. And we could do this in the lab and then pick this up, walk it out into the field, bolt it to the rocks, and then let the waves have their way with it, let the sun have its way with it, and so forth, and then record what those mussels are experiencing and how they're responding behaviorally to those changing conditions. So we did this out on a wave-exposed rocky point. The 2 little stars in the upper left there indicate where we put 2 of these groups of mussels on their plates. They were located sort of fairly low in the intertidal and then fairly high in the intertidal. So the picture on the right here shows me installing the low shore one, and then in the foreground, you've got the high shore one. And then they were left out there for multiple weeks. And so this is a quick time lapse on the left here, showing the tide sort of coming in and washing over the high shore plate. And the low shore plate is underwater at this point. You can't quite see it in the waves there. But we sort of allowed this thing to run and see what the animals are doing. I'm going to show you a little bit of data, but in animation form here. So I'm going to show you another sort of time-lapse video like we were just looking at, but there's 2 graphs here now. In the upper left graph, what we're looking at is data from that group of animals in the foreground of the picture, the high site animals. And on the upper right is a graph of data from the animals that are down lower on the shore. And what I'm going to show you is their gaping behavior. That is when they're opening and closing the shells. And so as you look up and down on the left axis, or the vertical axis on both of these graphs, you have the gape percentage. That is, how open or closed are they. If the values are low, they are closed up. If the values are high, they are open. And so as we start out our little video in the early morning with the rising sun, you see everybody's closed. They have very low gape values. The upper right graph is going to start opening up, or showing those animals opening up as that water level is rising. And then a few hours later, the high shore animals are going to start opening as the waves are splashing on them and starting to re-wet them. But you notice the high shore animals don't open all that wide, and then they start closing pretty early on here. The low site animals are still mostly open, and as the tide drops in the afternoon, we're going to see those low site animals on the right start to finally decide to close up as evening falls here. And everybody is closed again for the evening low tide. But looking at that, you see a whole lot more variation in that right-hand graph. Those low shore animals who spend a whole lot more time underwater during high tide have a lot more options in terms of when they're going to open, what they're going to do. They can open up, they can pump water, they can feed, they can breathe as well. The high site animals are really restricted in the amount of time that they have to do that same sort of behavior, because they're high on the shore. The waves are only washing them for a certain amount of time. And again, the fact that they don't open very wide is really a consequence of the fact that on this particular high tide that I just showed you, they weren't spending a whole lot of time fully submerged. They were really just getting washed by waves, and then as those waves retreated, they were back out in the air. So they would open a little bit, presumably get a little bit of water exchange, probably not doing much feeding. And generally what you'll notice is that animals, mussels in this case, that are living high on the shore, they don't grow nearly as fast as the animals that are living lower on the shore because they just have less time to eat. I'll show you the same sort of animation, but in this case with temperature. So recall again, we had buried temperature sensors in each of these mussels that we put on these plates. And it's going to be the same sort of animation with animal or data from the high shore animals on the left-hand graph and data from the low shore animals on the right-hand graph. This is the same day, the same exact time that I showed you in the previous animation. Notice in this case, in the left-hand graph, those high site animals that are out of the water are getting fairly hot, but there's also a lot of variation among the individuals. So each different color here represents a different individual mussel. Once high tide comes, everybody basically goes down to ocean temperature, was about 16, 17 degrees Celsius at this point. But notice again, the high site animals on the left, you see again, a big spread in terms of what the individual mussel temperatures are there. The low site animals are a little bit more grouped because they spend most of the day at ocean temperature. But there's, again, at the end here, a lot of difference in terms of the individual thermal experience of each of those mussels, particularly on the high shore. Some of them got upwards of 35 degrees Celsius. Some of them stayed down at about 25 degrees Celsius. And so that's a 10 degrees Celsius spread on a plate that I showed you the picture of where those mussels can't be more than about yay far apart. So a little bit of graph data. Really what I want to point out is on that far right graph, when we compare low shore animals and high shore animals, there is often a big difference in terms of the maximum temperatures that they experience. Those red dots representing high shore animals that are getting a lot hotter on most days. But their daily average temperature doesn't differ all that much between high and low shore. The minimum temperature doesn't differ all that much between low and high shore. And so really it's about those maximum temperatures during daytime low tides that we're seeing a lot of difference among these animals. The other thing I'll point out is that, again, that small scale variation. So I've highlighted two mussels here, the red and the blue ovals here. This, under the same set of conditions, on the same day, literally sitting centimeters apart, one of those mussels hit 37 degrees Celsius. The other one hit 23 degrees Celsius as its maximum temperature. So we have a 14-degree spread in body temperature for two animals that are literally just body lengths away from each other. And that's really a consequence of sort of their orientation, how much shading they have from neighbors and so forth. But it's telling us that in a bed of mussels that are all living sort of elbow to elbow, there can be a lot of individual variation in their thermal history or the amount of stress that each individual experiences. So I want to move from mussels on the open coast and start talking about oysters and mussels in bays and estuaries. And so what we've been doing a lot in recent years is sort of using oysters and mussels with those sensors that I showed you as effectively biosensors, telling us something about how the environment is doing and how the animals are responding to the environment. So we put this under the heading of our Habitat Heartbeats project, and I'll tell you why in a second. We get to work in some interesting places down in San Diego County, including this field site in the picture on the right. The reason we call it Habitat Heartbeats is that one of the sensors we've added since the early days is an actual heart sensor. So there's a very slow moving video there on the right showing a beating heart from a dissected oyster. So oysters and mussels both have hearts that beat, just like us. It pumps fluid around their circulatory system, just like us. And it means that just like we have Apple Watches or Fitbits that have little sensors that tell us what our pulse rate is, we can take those same sort of sensors, glue them to the outside of the shell of a mussel or an oyster, and have it tell us what the heart rate is of that mussel or oyster. So the picture on the right-hand side here shows one of my students holding a heart rate sensor that's essentially what is in an Apple Watch on the outside surface of a little mussel, and then on the computer screen behind her, you can see a pulse trace. And so these animals will sit there with a heart rate of 15, 20 beats a minute. Again, very, very similar to a human, although obviously a little bit slower. So these sensors, you can get them really quite small. The actual sensor itself is just this little glass windowed sensor here, and it's just shining a little infrared LED out of the sensor through the shell of the animal. That infrared light is bouncing off of the moving heart of the animal and being reflected back through the shell of the animal to the sensor, which has an infrared sensor on it. And so from that, we're getting that pulse trace on the right-hand picture there. And we can waterproof that. We can glue it to the outside of the shell and have it stay there for months at a time and continue to give us data for these animals. So we've done this in a couple of different places. In Newport Bay, there was a project, there is an ongoing project, trying to restore oyster reefs in the upper portion of the bay. So this is really led by collaborators at Cal State Fullerton, Daniel Zackrell, and folks from Cal State Long Beach, including Christine Whitcraft. And back in about 2016, they undertook a major project to transplant a bunch of eelgrass or seagrass around in the bay, and also put a whole bunch of old dead oyster shells in these middens or lumps there on the shore in the picture on the right. Now, the idea is they're putting dead oyster shell out there, but live oysters really just love to settle on other oysters if they can get a hold of them. And so the idea is to foment the settlement of natural oysters washing around within the bay on these hard oyster shells that are suddenly present in the bay. So, you can imagine Newport Bay has been really highly altered by people over the years, dredging, building marinas, and so forth. And so whatever original native oyster reefs were in Newport Bay are no longer there. And so now we've got people trying to rebuild oyster reefs in there, trying to encourage native oysters to settle, start reproducing, and start growing into self-sustaining populations. Our part of this was to actually put oysters on these reefs and look at whether they were surviving and how they were doing. So we, again, built little custom data loggers, in this case, in a size that we could shove into some plumbing pipe in the lower right there. I, of course, bought the clear stuff because it looks cooler. You can see the circuit board inside. So you've got to have that kind of sexy part to it. But we could then attach sensors to native oysters, like the middle picture there, take that entire pipe with the data logger inside of it, and go stick it out in the field. And stick it out in the field, I mean shove it in the mud, so that on the right-hand side, all you've got sticking out is the little tiny oyster with its little leads going back into the mud, into that pipe housing. And so we were putting these out in areas where there had been eelgrass restoration, where there had been oyster reef restoration, where there was just unrestored mud as well, and looking at the heart rate and the opening and closing of these animals. I'll just briefly show you that perhaps unsurprisingly, when it rains, when we get rain events like those blue bars on the top, what we see is among all of those oysters, everybody's closing up. So the black lines in the lower graph show that gape opening again, going down towards zero, because these animals spend a whole lot of time closed when there's fresh water, fresh rainwater flushing through the Newport Bay. So we do see these sort of coherent group behaviors when they've got major environmental changes, like a big freshwater storm coming through their environment. We're doing similar sorts of work at a couple of different sites in San Diego. And so I've highlighted here Los Penasquitos Lagoon, San Diego Bay itself, and then the Tijuana River Estuary. I'll talk about two of these as sites that we're sort of intensively studying. So Los Penasquitos Lagoon is a, of course, human impacted estuary. It's a photo on the right here. It's just a bit north of UC San Diego. It's just south of Del Mar. So it's prime real estate, of course. And it has a train trestle running through it. It has a road built along the beach there. And those two things are sort of fairly major alterations to the flow of water, particularly that road that cuts off the mouth. So I shouldn't say it cuts off the mouth. It spans the mouth and sort of restricts where the mouth of this estuary can go. But all the green area you see there is mostly undeveloped land. It collects runoff from the surrounding land above it, the watershed above it, and it funnels it out to the Pacific Ocean. But for most of the year, being that this is Southern California and it never rains, this is really an ocean-dominated estuary. Ocean water is getting into that little arm of the estuary that you see there, and the animals that live in there are marine animals, including oysters and mussels. And so we've been putting out moorings, floating moorings, like the central picture here, with oysters and mussels with sensors on them in that main channel, and looking again at how these animals are responding to changing water quality conditions in that lagoon. So we've got a group of oysters on the left there with their gape sensors attached to them, all the wires, the bundle of snakes and wires running back to a waterproof box. That waterproof box gets stuck up on top of a boogie board at the top of our central picture there. That is our float in this case. Gives us a nice flat floating surface to attach a box to. We then attach groups of mussels or oysters to the underside of the boogie board to sort of sample what the surface waters are like. And then we run wires down to another group at the bottom of that picture of oysters and mussels hanging off of a little PVC pipe there that are near the bottom of the estuary. And alongside of that, we've got a whole bunch of sort of normal water quality measuring devices, things that are measuring salinity, temperature, dissolved oxygen, chlorophyll, that is a proxy for the phytoplankton that are in the water that are the food for these animals. We dump this out in the estuary and we let it sit there for several months. And we occasionally have to go out and download the data and look at what these animals are doing. I'll show you a similar sort of effect of big rain events in this case. So I've highlighted in the picture on the left the animals at the surface that are attached to the bottom of that boogie board. And the graphs here, what we've got is at the very top, a graph of rain falling in the watershed where that empties into Los Penasquitos Lagoon. And then each of the red graphs below it represents opening and closing shell data for an individual oyster in this case. And what you see is at the very start of this graph, we've got two fairly big rain events. And you'll notice that with the blue arrows, I've highlighted periods where that gape value on the graph goes straight down to zero and stays down at zero. And the time scale here is such that we're looking at those under those blue arrows of about 4 to 5 days where those oysters are completely shut. They never open during that period. They are effectively holding their breath and starving themselves for that entire period because they're waiting for that fresh water to get through the estuary so that the salt water comes back in and sort of bathes them again in this kind of salt water that they want to be sitting in. If we look at the same sort of data for the animals that are just about 4 or 5 feet deeper in the center of that channel, again, here I've highlighted it in green, that lower set of animals on our mooring, and I'm showing you the same rain data at the very top, but now the green graphs represent opening and closing shell data for individual oysters at the bottom. We don't see that same prolonged closure period for the animals during those initial rain events. What you see instead is individual animals maybe closing for a few hours and then reopening. And so you do see gape values going from high to low more or less every day for these animals. But really they're spending most of their time open, feeding, pumping water through their gills, collecting food, exchanging oxygen, and so forth. So these animals that are just a little bit deeper in the water column are not experiencing as much fresh water. The fresh water, being less dense than salt water, tends to float at the surface of an estuary. So if you're an oyster down near the bottom, you're sitting pretty. You don't experience a whole lot of fresh water. You don't experience low salinity. It's not stressful for you, and you can just sort of stay open and keep feeding that whole time. If you're at the surface, it's a different picture during the rainy season. You might spend most of a week closed up, not doing anything useful, just sort of waiting for the water to get a little bit saltier so that you can go back to breathing and eating. Now, Los Penasquitos is a nice place to work. When we need to go out and service our instruments, we get in our kayaks and we just sort of paddle around and we go open our boxes and we download our memory cards and we fix the batteries and so forth. And it's really, fairly nice place. We pull the kayaks back up onto shore, we walk across the street and get tacos at Roberto's, and then we drive home. Really highly recommended kind of field work. The other place I'm going to talk about is the Tijuana River estuary. And here's some foreshadowing here, right? What am I going to say about the Tijuana River estuary? So the Tijuana River estuary is situated right at the US-Mexico border. So at the very bottom of our aerial photo here is that there's a fairly straight line. That is the US-Mexico border there. So the bulk of the Tijuana River estuary is on the US side of the border, all that green stuff that you see there. And then you've got the town of Imperial Beach is all the development just to the north of it. We work in a field site where that orange arrow is, very near to the mouth, where the mouth of the estuary, the Tijuana River, dumps into the Pacific Ocean. And again, it's a fairly oceanic sort of set of conditions just within the, near the mouth of that river. But the Tijuana River estuary has some challenges to it. So here we are zooming into the mouth right there, and we're going to sort of fly upriver here. We're going by a naval aircraft station there on the left, going up into the somewhat developed Tijuana River Valley. And we finish here just floating above what is the International or the South Bay International Wastewater Treatment Plant. The wastewater treatment plant is situated right at the border, and it exists because for decades and decades, as the growing US side and the growing Mexican side in terms of population were growing, there was a greater and greater amount of runoff going into the Tijuana River, and including a lot of sewage. And so the US and Mexico got together, worked on the Tijuana sewage plumbing, essentially, put pumps in place to pump sewage to this wastewater treatment plant. The plant processes the wastewater just like all of our normal wastewater treatment plants, sewage treatment plants, and then it sends the treated water through a subterranean pipe out into the ocean and dumps it offshore, again, just like we do up here. And so ideally, there isn't sewage getting into the Tijuana River, ideally, right? Pumps break down once in a while. Being a sort of federally managed project, there's chronic underfunding, chronic lack of preventative maintenance. And so the sewage treatment plant is not always working to full capacity. And when it's not, that means there's sewer water going into the Tijuana River. So instead of getting in a kayak and sunning myself and make sure I've got lots of sunscreen on, I'm on the right there in a full body dry suit with double gloves taped on so that I don't touch that water as much as possible, wade myself out to our, again, mooring out in the channel and do the servicing that I need to do. And then come back in and disinfect myself and try and again, not get anything in my mouth and my eyes and so forth. And it's not the most pleasant place to work. Now, if you've spent time in an estuary, you know that often at low tide, they don't smell that great to begin with. It smells the same here, but you kind of suspect that that stink is not just a naturally derived stink from the anoxic soil. It might actually be from other sources. So we do the same sort of thing. We put animals out and look at how they respond to the changing environment. So there's a lot of data here. I'm going to walk you through it. I'm going to highlight and zoom in on 3 particular days here and sort of show you what's going on. So on the top graph, again, we've got rainfall. So where you see those spikes, that means rain was falling. Natural rain falls into the river, the watershed around the Tijuana River, flows through the Tijuana estuary. That water is mostly clean. It's not sewer water. And the animals, may have to respond to it because it's fresh water as opposed to salt water. In terms of what we see for the animals, the lower 2 graphs I'm going to highlight here. The second to the bottom is the gape opening. So again, when the shells are opening and closing, high values mean the animals are open, they're kind of happy, they're feeding, they're breathing and so forth. Values near 0 mean they're closed up and they are avoiding that water. The bottom graph shows us heart rate from those same animals. And so you sort of see the trace go up and down through time. And in these blue boxes, these blue vertical columns that I've highlighted here, we see 3 different days right around midnight, right during low tide, followed by a lower high tide, followed by a higher low tide, where really the influence of the ocean is being overshadowed by that fresh water flowing out. And what we see is the animals all closing up. So in that second graph, we suddenly see them going down to 0. And just below that, we see the heart rates all drop. So you might imagine you clam up as a mussel or an oyster, and you're no longer exchanging water. You're no longer feeding. You're no longer getting oxygen from the environment around you because you have isolated yourself from that environment. And so is there really much value in continuing to pump your heart and pump blood around inside your circulatory system? I would argue no, because you've used up whatever oxygen is going to be in that, that circulatory fluid pretty quickly, and you're just burning energy pumping deoxygenated blood around in your body. So these animals drop that heart rate down to 3 or 4 beats a minute. And in some cases, they're probably basically just stopping the heart for minutes at a time, not bothering to contract it at all. And then once in a while, they'll contract it and move a little bit of fluid around. When the tide rises, in our graph second from the top, we see a brief respite. So all these animals sort of reopen very quickly for a brief period of time, do a little bit of pumping. We see their heart rates jump up a bit. And then as that tide drops again and the freshwater influence comes back, they close up again for multiple hours, they drop their heart rates. And then ultimately, when the higher high tide comes, in the early morning in this case, that ocean influence, salinity, salt comes back, they taste that somehow, open their shells, start pumping water, go back to normal. We see this happening day after day after day with these big drops in heart rate, presumably conserving energy and trying to sort of make it until the water conditions go back to something more favorable for them. The Tijuana River, as I said, has that issue of occasionally having nasty stuff flow through it. I'm showing you data here from about the year 2000 through 2024. And in yellow, I've highlighted periods where there's basically no river flow happening because it's the dry season, the sewage treatment plant is working, and there's not a lot of normal runoff going into the river. The blue spikes that you see along the way are mostly rain events. So there's natural water flowing through the river. But I'm going to zoom in on the last couple of years here in the green box on the right. And now we've zoomed into the period 2022, 23, and 24. You see some yellow zones here with low or no river flow. But then in early 23, you just stop seeing yellow. You see basically at least some consistent level of maybe low water flow going through, even during the dry season when there should not be any fresh water entering that river. And so what we're seeing here is a signal of the pumps breaking down and water, untreated sewage overflowing into the river and flowing downstream, creating this record of river flow when there kind of shouldn't be. That's led to some fairly dire consequences for the surrounding human population. I should say dire consequences for the economy of the surrounding population. Because in the bottom of our graph here, there have been over 1,000 days of continuous beach closures right down by the Tijuana River estuary. That is adjacent to that town of Imperial Beach. And that means bad things for the economy of Imperial Beach, which should have surfers coming to it and beachgoers coming to it and people going on vacation. But when you can walk out on the beach and you can get that whiff of, who knows what, it's a bad sign. So it doesn't do well. And what we see in terms of oxygen in the water is particularly telling for when we've got bad conditions here. So on the left, I've got data from 2020. Notice that the oxygen, the green line, is bouncing around between 8 and 6 milligrams per liter. That's what we call sort of normal oxygen levels in normal estuary water. In 2024, I've got a period here where we had that continuous river flow and presumably pollution going through the river. Notice that that oxygen picture is basically flip-flopped. It's spending a whole lot of time at essentially zero oxygen with these little brief spikes of oxygen, which are really being driven by the ocean water being able to get back in at high tide and re-oxygenate the animals. And so that's been really bad news for the animals. These are some gape data, shell opening data for oysters and mussels from this last summer when there shouldn't have been any river flow because it was the dry season, but we were definitely seeing river flow. And at the top, we see 3 of these oysters all managed to die. One of our mussels managed to die. A couple of them managed to survive during this period, but it's been really bad for these animals down there. The other thing we're doing is trying to integrate sort of real-time data reporting. So we've got data loggers now that talk to the cell network and tell us things about what is going on with individual mussels. So we've got a website there at the top showing us data, continuous updates from individual animals, whether they're open or closed. And so we've got this happening in Los Penasquitos, we've got it happening in San Diego Bay. We need to put our animals, live animals, back out in Tijuana and hopefully get live data from there as well. And hopefully this is the sort of information that will eventually be useful to managers as well as sort of a canary in the coal mine for when things are getting bad in the environment. So at the very end here, I'm just going to return briefly to the title of this talk, which was Prying into the Private Lives of Mussels and Oysters. And I'm going to say there's a bit of a double meaning here. I'm going to return us back to this intertidal set of Mytilus californianus on the wave-swept coast. I'm going to show you another one of these little animations. This is actually just a few minutes worth of data. And I'm going to show you some data that include the pink line being the shell gape when they're open or closed, and the yellow line being acceleration, sort of when this animal is being knocked around. And so we start it running here. We see they're closed. The pink line is at the bottom. Suddenly the pink line is going to open up, go quite a bit higher. And you see a whole bunch of acceleration, that yellow line bouncing around. So what is happening here? What is happening here, you may or may not have noticed that in the foreground of our picture here, there's a little guy standing on our bed of oysters, or sorry, bed of mussels. That is an oyster catcher. An oyster catcher likes to go around and eat mussels that it can find, despite the name. They should be mussel catchers, but in this case, they are called oyster catchers. And what that oyster or catcher was doing was going around and looking at our plate of mussels that we'd put out for it and saying, oh, this looks like a good one, jabbing its beak into it. And here's a sort of three-dimensional reconstruction of those acceleration data in space, showing you that this poor mussel was being wanged all over the place as this oyster catcher was jabbing its beak in there, trying to open that shell up and actually scrape all of the meat out of there and ultimately eat our mussel. This happened to us 3 different times for animals that were fully instrumented, so we have sort of records of this happening. And in hindsight, it makes complete sense. Of course, we've put big, fat mussels high in the intertidal where they aren't normally, and the oyster catchers are going, this is the greatest thing on earth. Why would I go eat anywhere else? Why would I eat natural mussels when I can just eat these things with the wires on? So what we managed to catch here was a version of essentially what these oyster catchers are doing a lot of the time. So they will eat things like limpets and they will knock other stuff off the mussels. But a lot of times what they're really looking for is mussels that they can get their beaks into. And they have to do it during a very narrow window of a returning tide or a descending tide when the mussels are still sitting a little bit open. So they can't get their beak into a fully closed mussel, but they can get their beak into a mussel that has started to open because, ooh, it felt the wave splash, and maybe it's time to reopen and start breathing and eating again. But in this case, this oyster catcher, now this is sped up at this point, has managed to get its beak in there. It manages to sort of force that shell open, and now it's basically just sitting there scraping around on the inside of the shell. We'll let it run here for a few seconds, sped up, and then it'll slow down again for a second. As we slow down, you're going to see it actually pull the entire innards of the mussel out of there and just very deftly pull that thing up into its mouth, swallow it, and at that point it'll get up and move along and go look for another mussel that has made the mistake of sitting a little bit open when the oyster catcher can walk up to it and grab it. So there is our second meaning for prying into the private lives of oysters and mussels. So I want to finish just by acknowledging a whole lot of people that have been involved in various parts of this work. Folks at San Diego State, again, Scripps, the Tijuana River Estuary, Fullerton and Long Beach have been important collaborators for that Newport work. And then my collaborator up at Washington State, who sort of got me started down this road with the mussels in the open shore, and a whole bunch of funders that are involved in various parts of this work as well. So at this point, I will take questions, and I thank you for your attention.
I just want to say this is one of the greatest talks on mussels and everything else I've heard. And I'm thinking it was in March of 2020, there was a red tide that came in.
Yes.
And I was wondering if you had any data that
We certainly do, actually. It was very fortuitous that we had animals out in the field and actually in the aquarium down at Scripps. And so the Scripps aquarium system, portions of it got hit really hard because they pull that red tide water into their system. And it was, their system is filtered, but whatever was toxins were in that water were still making it through the filter system. And they had a lot of die-offs of animals in their systems. We had, in that case, oysters with game sensors on. We watched them sort of close up and sit there because they tasted what was in that water and they didn't like it. We also had animals that we threw out in Los Penasquitos lagoon, the sort of cleaner one that I talked about first, that had a bunch of that red tide water flowing into it. And for like the worst possible timing in the world, every year the state parks goes and cleans out, sort of opens the mouth of Los Penasquitos lagoon. In order to do that work, and removing sand around the beach to do that. But in order to do that, they actually create a sand berm to keep the water from flowing out of the estuary while the tractors are running around. Well, they did that the days after that red tide water had flowed into the estuary, trapped all that red tide water in there, and there were mass die-offs of all sorts of animals in Los Penasquitos lagoon, including a bunch of our oysters and mussels, where again, I sort of showed you that graphic with the little skulls on the we had that same sort of thing where we just day after day we're watching individual animals just sort of drift open because they have died, and that resilient hinge in the hinge of the oyster just sort of forces that shell open. This is why when you go buy oysters and mussels, you want the ones that are very tightly closed. You don't want the ones that are kind of laying lazily open, because those are the dying or the dead ones. So yeah, we saw a big impact of that. At that point, it was basically a month-long red tide down in San Diego.
Yeah, they killed all the mussels up in Rio del Rey and King Harbor.
Yeah, that was an impressive red tide.
So the last sequence that you showed us is clearly a testimony to the intelligence of birds, and I'm all about birds. What I would like to know from you is, how long did it take for the oyster catchers to actually realize there is a smorgasbord out here? Do you have any idea?
So I mean, we had those plates of mussels out there for about 2 weeks. I'm trying to remember exactly, but it was partway through that deployment that the first one got nailed, and we didn't know what caused it initially. It was later on during that 2 weeks that I had actually put that little time-lapse camera out there and happened to capture what was either the second or third predation event. So it took them, days, 7-10 days to actually figure out that, oh, these.
And then they kept coming back.
And then at least the one oyster catcher came back multiple times.
So, okay, so oyster catchers are usually in groups, so there's just one thing.
So usually in pairs, I say one without actually knowing them. At that site, we had a resident pair that would cruise around. I'm guessing that it was the same one that would come back day after day. I don't know that for sure, but reasonable chance, because it is usually the same pair that we would see out there. So maybe it was one or the other of that pair, but they figured it out. They came back for more. Yeah.
I'll be a little greedy and ask 2 questions. For your experiment with the scallops, when you use the laser, did you have to ping that syringe at the same force every time, or did you just take an average of the amplitude?
So the goal was to ping it as close to the same force as we could every time, but we were then basically really interested in what the frequency of the decay of that twanging was. And so in some sense, didn't matter exactly how hard we hit it. We just needed to hit it hard enough that we got a good starting bounce, and then we just sort of watch it decay.
My other question is, with the restoration projects, is there any effort into working with indigenous communities around there to help restore those areas?
So that project, I don't actually recall if the local tribe groups were involved in that. Partly because I'm not the right person to ask for that, because I wasn't involved in 2016 when they were putting that stuff out. So I don't know off the top of my head.
I'm fascinated by the temperature differences that you saw in the mussels that were very close together. Do you think that in the wild mussels that are not glued onto a plate, that they're doing clever things to avoid overheating?
No, I think there's still a fair amount of variation among natural mussels as well. So that sort of general idea of looking at multiple mussels within the same bed has been repeated in a couple of different guises. In some cases, using silicone-filled shells with temperature sensors inside them shoved into naturally attached mussel beds. In other cases, taking those thermocouple probes and shoving them into live mussels sort of temporarily. And there's basically, I think, going to be sort of winners and losers in a mussel bed, determined in large part by where exactly in that bed they happen to be. So if they're down deep with a whole bunch of neighbors around them, they're going to be pretty well shaded. They're going to be pretty well covered from the sun. But there's the downside that if they are down deep in that mussel bed, they're not as exposed to the water with all the food flowing over them, whereas the animals that are up on top are getting a full flow of water and food over them. So there's probably trade-offs involved there in terms of how risky do you want to be in terms of thermal stress versus getting a whole bunch of food if you're the one that's out there exposed.
So when you're done wiring these critters up and you've done the experiments, you take the wires off?
So yeah, we can. It's sort of peeling the super glue and epoxy off of the shells, and we can absolutely do that. I will say that in a lot of cases, the animals are dead by the time we pull those wires off. Because especially in the field, we leave them out for as long as they're going to continue collecting data. And so eventually we start to see that gate signal of them just sitting open the full time. So we go out and say, oh yes, this animal has died. So we will take it back in and eventually replace it with another live one. Larry?
Yeah, I was wondering, always I've been wondering, why do they call them mussels?
Almost, I don't know.
Oh,
It's an excellent question. I've never looked into why that is. I guess just to be confusing, given that these mussels have muscles inside of them, the muscle physiologists work on because the muscles can be super interesting because the catches stay closed for hours on end without a whole bunch of energy being burned. The whole thing closed. Life expectancy, so a decade or more for both the mussels and the oysters. And I don't know that we really have a good handle on what the maximum life expectancy is for an animal, especially if you're holding it in sort of ideal conditions where it's protected from predators. But easily a decade for either of those species. Michael?
So you showed a lot of opening and closing of these in response to what was going on in their environment. Do you know their detection? How do they detect that it's a good time to open?
Right. So how do they decide, when it's time to reopen? Oftentimes, we imagine when it's a low salinity signal, these animals are not perfectly hermetically sealed. And so it may be the case that there's still small amounts of sort of exchange with the outside water to which they can sort of taste the salinity of that water. Same sort of thing may be true for oxygen as well. Oxygen permeates through the air gaps around the shell. And so that's probably what's happening. In some cases, we will see sort of brief little opening testing sort of behavior, but it's not necessarily, but during those prolonged closures, multi-day closures, we didn't see that. So they probably are somehow sensing still, in fact, salinity is basically at zero, it's fresh water without having to.
Where are they, the people consume these, where is the biggest quantity of mussels and oysters that people consume, and how are they harvested?
So in terms of human consumption, certainly there are aquaculture operations that are growing. When we're talking about oysters you're seeing on the food market, those are almost entirely going to come from aquaculture operations. On the West Coast, most of those are happening from Morro Bay northwards. Tomales Bay up in Bodega Bay is a big one, up into Oregon, up into Washington, and around Vancouver Island.
What is that process like? What do they?
So how do you grow an oyster? What they do is in their hatcheries onshore, they will breed oysters. They will basically trigger them to spawn. They will have the baby oysters forming growing within the lab setting, and then they will take those once they're somewhere between half an inch and an inch long, put them into little mesh baskets, and then put those out in the water in the bay or in the ocean. And in that case, I think in the West Coast, most of the operations are sort of hanging those plastic mesh baskets on racks, so that they're sort of floating around a little bit. And the goal is actually to not have the oysters attached to anything like they would naturally like to do. The goal is to have them kind of tumbling around. They call it tumble culture because that tends to actually force the shell to grow deeper, so there's more room inside for more meat that makes it more saleable oyster. So that's where the majority of that's coming from. Mussels in this country are also going to primarily be coming from aquaculture operations as well. And this is probably sort of true. And you can grow mussels attached to ropes where they will sort of attach their viscous threads and then hang those ropes out in the water and same sort of thing, let them grow for a couple of months, a couple of years, and pull them off the ropes and sell them off the market. What do mussels eat? They eat phytoplankton. So they eat little green sort of algae that are floating around in the water that are so small that you need to look at them under a microscope to see them.
I also like rotifers.
They also like rotifers, yeah.
You ever have the end of the year with your compatriots, a big feast of mussels. That capital.
So I try to convince people that we should eat the ones out of Tijuana estuary. So I still eat oysters and mussels, but I eat the ones that have, agriculture. And, that's sort of my bent towards food safety, I suppose, is getting them from growers to sort of know what they're doing. I don't trust me to know what I'm doing, I'm crazy and healthy.
The water when we were like, heating up the water with the scallops.
Oh, how hot, yes, when we're testing their scallops behavior in Antarctica under different water temperatures, we raised the temperature up to, 16 degrees Celsius, which is sort of normal mid-year temperature up in Central California. So we took them real warm compared to what they would ever see, and they basically just become floppy, useless messes at that point. So there are lots of different things that like to eat mussels. Octopus likes to eat mussels. There are a variety of different snails that like to eat mussels. When the mussels are small, there are lobsters that like to come in and crunch them up and eat them. When the mussels are small, crabs will try to crush them and eat them as well. So yeah, there's a lot of different things that eat mussels.
Let's do a question from home.
Sure.
Okay. So thanks, Alfonso. Tamara asks, for the high shore animals, what advantages do they have in that type of environment? It seems like a more dangerous environment due to the higher temperatures, etc.
It's an excellent point, and we think that really what's happening there is that they're probably up on the high shore sort of as an escape from predators. So one of their major predators that I did mention, sea stars. Sea stars don't like to get hot on their way out, and so sea stars nibble around the bottom and the lower edge of the mussel bed, but they don't make their way up to the top of the mussel bed because they move slowly and might get caught during low tide, sitting out in the sun, baking and dying. So the thought is probably that living up high on the shore, even though it is more stressful for the mussel, in terms of the environment, it may actually be a little bit safer in terms of avoiding predators.
Let's do two more, and then I want to hear about that logo, Habitat Heartbeats.
No, okay.
You study how much or how far they moved over their lifetime, and is there any correlation with the temperature? Are they elbowing for better position to not heat up, things like that?
So the mussels, when they settle as little babies, and they're just a millimeter or two long, they're relatively mobile. They will actually stick their foot out and walk themselves around. And so there's definitely data showing that young ones, in some cases, as they're sort of juveniles, let's say, not babies, will move up into the upper levels of the mussel bed, presumably to actually get access to food. And so there are often sort of multiple layers to mussel beds, where the big ones that have been there a long time and can't easily release themselves and move around are sort of stuck down at the bottom, and the upper layers tend to be younger, smaller animals.
Have you put local scallops in cold water or in hot water and see how they react?
No, we have not. There are other folks who have done similar sorts of experiments predating what we ever did. I mean, the idea from ours was really coming from people working with tropical scallops and temperate scallops, like around, these sorts of waters. And so what we were adding on was, well, let's go get the cold water ones and see what they do.
All right, everyone, please join me in thanking Dr. Luke Miller for a fantastic talk.